Okay, good morning, and I first would like to thank the organizer for having invited me. As you see, I have had in the past some conflicts of interest because some of my research was funded by Nestlé and Ajinomoto, but most of it was funded by the Swiss government, by the Swiss National Foundation. I am Swiss, but unfortunately I have no conflict of interest with the Credit Suisse, but maybe it will come. No. Okay, so you already understood that there is a lot of controversy regarding whether obesity is linked somehow to an increased sugar consumption. And that stems basically from two kinds of observations. First, there is some parallelism between the consumption of sugar and the evolution of obesity over the years, some parallelism. And second, there are a lot of animal studies showing that putting rodents on a high sucrose or high fructose diet can cause dyslipidemia, can cause non-alcoholic fatty liver disease, insulin resistance, diabetes mellitus, even sometimes high blood pressure. But you have to be very careful with these animal studies because these are done with very high sugar consumption, and they are usually associated with some degree of hyperphagia and the development of obesity. Now, what do we know about sugar effects in humans? So first, what is different between sugar and other carbohydrates like starch? Well, sugar is made up of one molecule of glucose linked to one molecule of fructose. So when you eat sugar, you will absorb finally glucose and fructose, and the major difference between starch and sugar will be fructose. I won't go into the detail of the metabolism of glucose and fructose, but you see here the metabolism of glucose in hepatocytes. And you see that glucose is transported through glucose-specific transporters, and then it will be metabolized through glycolysis, and finally it will end up as either CO2 or glycogen. For glucose, only part of the glucose is taken up by the liver. And the glycolysis is very, very tightly regulated, first because this is an insulin-dependent process, and second because there is a feedback of ATP and citrate on glycolysis, though it's controlled by the energy status of the cell. As a result of this regulation, only part of the glucose will be taken up by the liver, and only part will be degraded to CO2. When you look at the metabolism of fructose, fructose is very similar to glucose in terms of molecule, and it's transported into the liver with the same glucose transporter. The difference is in the three initial enzymes. Fructose is converted first into fructose 1-phosphate by a specific enzyme, which is fructokinase, and then in trioses phosphate through the enzyme called aldolase B. And then there is a third enzyme, which is triokinase, which converts glyceraldehyde into glyceraldehyde 3-phosphate. This difference in only three enzymes has very important consequences because these enzymes have a very high affinity for fructose, and they are not regulated by insulin, and there is no feedback of the energy status of the cell. So virtually all the fructose which reaches the liver will be extracted, converted into fructose 1-phosphate, and then into trioses phosphate. When you eat an important amount of fructose once at a time, that causes a large synthesis of trioses phosphate, and these trioses phosphate kind of, let's say, invade glucose metabolism. So they outpass the capacity of the cell for oxidation, and they will be reconverted either into glucose or into glycogen, or they will be released into lactate. And finally, if there is a large amount of fructose eaten, part of it might be converted into fatty acids. Okay, when you look, we don't know exactly how much fructose goes into the different pathways. But when you look at the effect of a single fructose load, a pure fructose load in healthy resting humans, approximately 50% recirculates as glucose within the next 6 hours. Approximately 50% recirculates as lactate, and glucose and lactate then can be metabolized by extrahepatic cells. Probably about 15% gets into glycogen, and the remains is either oxidized within the liver or converted into lipids to be stored into intra-hepatic lipids or to be secreted as VLDL triglycerides. So there is a small conversion of fructose into lipids, but that's really a minor pathway. Now, I will show you a couple of results regarding short-term human studies with very high fructose or sugar intake. Just to get an idea, what is the sugar consumption, what is the fructose consumption in our populations? These are American data, but in the States, roughly 20% of total energy can be attributed to sugars, and that means 10% total energy as an average can be attributed to fructose. There is, of course, some variation, and maybe 10% of the population are really high sugar consumers, and they have fructose intake up to 15-20% of total energy intake. Now, I will focus on two specific aspects: the effects of high fructose feeding, short-term high fructose feeding in humans on plasma lipids and intrahepatic lipids, and second, on glucose homeostasis. There are a lot of studies showing that a high sugar diet or high fructose diet increases blood triglyceride concentrations. And what the study I show here is a study from my lab, but there are many studies before having shown that. What is shown here is that when you overfeed people with about approximately 15% energy as fructose over 4 weeks, you get a slight but very significant increase in blood triglyceride concentration. And that's essentially due to an increase in VLDL triglyceride concentration. What is interesting is that it remains sustained throughout the overfeeding period. Now that's overfeeding, though that's 15% fructose in excess of energy requirements. The mechanism for this increase in plasma triglyceride are not completely understood. First, there was an increase in fasting triglyceride and an increase in day-long triglyceride, or both fasting and post-prandial triglyceride. For fasting triglyceride, there is clearly a relationship between a stimulation of hepatic de novo lipogenesis, so conversion of carbohydrates into lipids. And that's what you see here on these slides, with a 6-day overfeeding with 30% energy as fructose. You get an increase in fasting triglyceride on the left, and in fractional hepatic de novo lipogenesis on the right. Now, there is one additional mechanism, is that it increases postprandial triglyceride. And on this slide, you see the lipid profile in volunteers having ingested a mixed meal together with either glucose or fructose. And you see that with the glucose meal, the plasma triglyceride concentration are lower, and that with the fructose meal, it is higher. The most likely explanation for this is that the clearance of triglyceride-rich particles is increased with glucose, but not with fructose. So both the stimulation of de novo lipogenesis and then lower extrahepatic clearance of triglyceride may contribute to this increase in, in triglyceride levels. Now, how much fructose do you need in your diet to have effects on fasting or postprandial triglyceride? This is controversial, but I compiled here a couple of studies having used different amount of extra energy as fructose, and you see that the fasting plasma triglyceride concentration starts increasing when the fructose content in the diet is 15% or higher. So that's also the threshold which was reported with meta-analysis, showing that when fructose consumption is higher than 10%, then you start having effects on plasma triglyceride concentrations. 15% is a lot, but that still is relevant to fructose consumption of a large portion of the population. Now, there is also concern that fructose may cause non-alcoholic fatty liver disease and then contribute to hepatic steatosis and possibly to the pathogenesis of non-alcoholic steatohepatitis. Now, we don't know much about the effect of fructose on intrahepatic lipids. What I show you here is a couple of studies done in my lab in which we measured intrahepatic lipid concentration with magnetic resonance spectroscopy before and after short-term overfeeding with fructose. And you see that when you add 15% energy as fructose, you don't see any significant increase in hepatic lipids. With 30 or 40%, but it becomes really very high intakes, then you see an increase in intrahepatic lipid. So clearly, fructose can increase intrahepatic lipids. Now, the units here are in millimoles per kilo. If it was expressed in volume percent, that would be something from going from 1% basal to 2.5% with the highest doses. So you get an increase in intrahepatic lipids, but it's much less than what is observed in obese patients, which is rather in the 5 to 10% range. Nonetheless, there is some concern that in the long term, this accumulation of intrahepatic lipid may be deleterious, because there are a lot of animal studies and cell culture studies showing that fructose, in addition to stimulating de novo lipogenesis and favoring lipid deposition, may also trigger some inflammation or may generate some toxic mediators triggering fibrosis. So that's a real concern, that in the long term, the effect on hepatic triglyceride may change, or rather the consequences of hepatic triglyceride may change. Now, let's get back to these human short-term studies. Is it an effect of fructose, or is it just an effect of energy? And here you see the effect of short-term overfeeding with 30% excess energy as fructose, that's the blue bar. And again, you see this increase in intrahepatic lipids. The same overfeeding, but with saturated fat, and you see that you also have a significant comparable increase in intrahepatic lipids. And finally, with 30% glucose instead of fructose. The increase is a little bit less, but it's also significant. So clearly, energy excess and not only fructose can lead to an increase in intrahepatic lipids. And similar observations were recently reported by a British group. That's a study which is available online, but which is not yet published. And in this study, 32 overweight males were studied. They were randomized to receive fructose or glucose, and they were studied on 2 occasions. Once after 2 weeks on a isocaloric high fructose diet or high glucose diet with 25% energy as fructose or glucose, and a second time with hypercaloric high fructose or high glucose diet. So same amount of fructose or glucose, but once as a weight maintenance diet, and a second time as an overcaloric diet. And what was reported was that in the isocaloric diet, there was no change in blood triglyceride concentration and no change in intra-hepatic lipids. While in the hypercaloric arm, there was a significant increase in blood triglyceride, a significant increase in intra-hepatic lipid, but the effect of glucose and fructose was the same. So it looks like the effect on intrahepatic lipids and plasma triglyceride may be more related to carbohydrate overfeeding than to an effect of fructose per se. Now, one question one may ask was that obviously when you have a high fructose diet in healthy subjects, you can increase plasma triglyceride, you can cause some accumulation of fat in the liver, but is fructose always deleterious? And here I retrieved an old study by our chairman who almost 20 or 30 years ago followed the 20 years ago, followed the Tour de France, let's say, which is, as all of you know, a rather strenuous physical activity. I never asked him if he followed that by car or with a bike, but I trust he did that by bike. What he reported was that, of course, the energy expenditure of these bikers was very high. It peaks to almost 2.5 times the normal energy expenditure. Their energy intake was also very high, and they had a very high intake of carbohydrates and a very high intake of sugary drinks or sugary foods. But I think that these people most likely are not at a very high risk of metabolic syndrome. So most likely there is more than just fructose. And we recently conducted a study in which we compared the effect of a high fructose diet with and without exercise on plasma triglyceride concentration and de novo lipogenesis. So this time, that's healthy volunteers. They received 30% of their diet as fructose, but as part of a weight-maintaining diet. And after 6 days, they received a meal with 13C labeled fructose, and we measured 13C palmitate in the blood as an index of de novo lipogenesis. What you see on the top left is that after this 6 days of a high fructose diet, there is a slight increase in plasma triglyceride concentration, but which is significant. And on the top left, you see that there is a marked stimulation of hepatic de novo lipogenesis. Now, when you repeat the same thing in the same subjects, but by adding 2 times 30 minutes cycling in the day and adjusting the dietary intake to maintain energy balance, you see that you completely restore plasma triglyceride concentration, and you completely prevent the stimulation of de novo lipogenesis. So clearly, not only the dietary intake, but also the physical activity and possibly other factors in the environment will modulate the effect of fructose. Now, I'm done with the effect of fructose and lipids. Very quickly, in animal studies, animals usually develop insulin resistance and diabetes. And you read you read almost everywhere that it is well known that fructose or sugar causes insulin resistance. Now, if you look at the real evidence, that's pretty slim. Here you see that that's a complicated slide in which we overfed subjects with fructose with or without fish oil, but we will focus only on the control condition, that is normal diet, and then, which is the white bar on the left, and the high fructose diet, which is the solid bar in the middle. And here you see hepatic glucose production in basal condition, fasting, and you see that after a high fructose diet, there is a slight but significant increase in hepatic glucose production. Now, the right part of the graph are endogenous glucose production measured during a hyperinsulinemic euglycemic clamp, so during insulin infusion at a low insulin infusion rate and high insulin infusion rate, low at 90 minutes and high at 180 minutes. In control subjects, you see a very marked suppression of glucose production, and with fructose, you see that the glucose production is less suppressed. And that clearly shows that the liver is less sensitive to insulin after this high fructose diet. Now, when you look at total glucose disposal during this same experiment, which is reported on the lower graph as glucose infusion rate, GIR, you see that the total insulin-mediated glucose disposal is absolutely the same. So there is some degree of hepatic insulin resistance, but there is no whole-body insulin resistance. At high insulin, it is generally admitted that insulin-mediated glucose disposal reflects mainly muscle insulin sensitivity, and that's fair evidence that fructose by itself will not cause muscle insulin resistance, at least not in the short term. Now, there are some alarming reports showing that fructose overfeeding causes more alterations of glucose homeostasis than glucose overfeeding. And here you see studies from the University of California at Davis, from Kimber Stanhope and Peter Havel, in which they overfed subjects during 10 weeks. They were overweight subjects, and they were overfed during 10 weeks with 55 to 30% total energy as either fructose or glucose. And what they reported was that on the left part of the graph, people overfed with glucose did not change their glucose tolerance, and people on the right side who received fructose did have a significant increase in postprandial insulin and glucose. Now, if you look at the threshold at which we would talk about impaired glucose tolerance, which is reported in red, you see that the difference between the two groups is not really tremendous. And this slight increase in postprandial glycemia may possibly be accounted for by changes in hepatic insulin sensitivity. So I will come to conclusion. And first, regarding dyslipidemia, I think that there is good evidence that fructose can cause an increase in fasting triglyceride concentration, that it is dose-dependent, that it might be due to both stimulation of de novo lipogenesis in the liver and to an alteration of the clearance of triglyceride in extrahepatic cells. I didn't show you that, but it also changes a little bit the nature of LDL, and it increases small, dense LDL particles, which may confer an increased atherogenic risk. But these effects are observed at relatively high dose, and they are largely prevented by exercise independently of energy balance. Regarding hepatic lipid accumulation and non-alcoholic fatty liver disease, here again, at high doses, fructose increases intrahepatic fat. This effect looks to be more related to excess energy than to fructose per se. The short-term effects lead to much lower accumulation of lipids than is observed in obese patients or in patients with the metabolic syndrome. But there is nonetheless a real concern that in the long term, fructose may also trigger some degree of inflammation and possibly trigger the switch from non-alcoholic steatosis to non-alcoholic steatohepatitis. And finally, fructose as such has little effect on glycemia. A high fructose diet has little effect on glycemia. It slightly decreases hepatic insulin sensitivity. I don't know the meaning of that, whether it's just an adaptative change to a high carbohydrate diet, whether it has pathological significance. In the short term, it does not decrease whole body insulin sensitivity, but the long-term effect remains to be assessed. And finally, I would end this talk by saying that there are a lot of many questions to be answered. First, there is this slight increase in plasma triglyceride concentration with very high sugar intake. Is it significant in terms of risk of heart disease? That's something which remains to known. The effects on hepatic lipid and on the development of non-alcoholic steatohepatitis remains to be known. Long-term effect on muscle insulin sensitivity remains to be known. But what I really believe is that when you don't see short-term effects, and you call short-term 10 weeks, I would say that long-term effects are more likely to be related to changes in body composition than to really long-term effect of substrates. So I guess the real and most important question is, does it increase energy intake and does it cause obesity? And I will end here, and I still want to thank all my collaborators. Here you see people in my lab who did all the work. I would like to thank my colleague Bush and Kreis in Bern, who did the magnetic resonance spectroscopy for us, Jean-Marc Schwartz at UCSF, and colleagues in Zurich, Oxford, and St. Louis, who all contributed to this work. Thank you for your attention.